when you think you have.
To start?
Yeah.
Okay. Put this thing. Yeah, put the slides up.
So CME, I was going to introduce yourself.
Okay, yeah, so.
What's the name?
What's that?
Someone of.
Oh, somebody just moved. Okay, so I'm Eric Hartz. I've been working at the JCRC for a lot of years. And so if you don't know me, I'm sorry. It's okay. That would have been annoying though. Just because it is. Okay, so I'm just going to present today on a collaboration that Western University and the University of British Columbia and a number of other partners at University of Montreal in Canada are working together with the JCRC. And the purpose of this collaboration is to bring an HIV cure to Africa. That would be the overall objective, but the bigger, I think, more important aspect is to provide capacity building in the JCRC in particular to advance therapeutics, HIV cure initiatives, and gene therapy with the goal of providing the capacity for production at a good manufacturing practice, production for clinical use, and to provide abilities to do therapeutic monitoring at point of care sites rather than centralized if we can help it, because HIV cure, that's going to be required considering how many type of tests you will need to do early on in that treatment. And then the other thing is to develop the network between the JCRC and Western University and all our partners so that we can do this stuff simultaneously. So as I'll indicate, we have a circumstance in most low to middle income countries, low resource settings, that there's somewhere between a 5 and an 8 year delay between the most advanced treatments for HIV and most diseases happening in the high income countries before they're provided to the low resource settings or low income countries. And that has had major implications in HIV. We're finishing up a study now that has shown that DTG based treatment could have actually been much more effective if it was administered at around the same time as it was in high income countries, rather than watching a lot of NNRTI based treatments fail. So these are the types of things that we're trying to advocate for. And we have a large grant, so I'll present on that now. Okay, well, he's trying to put up the presentation. So what I'll go through in very lay terms, what is the therapeutics that we're developing, how we're going to produce them, both here and at Western University. That I'll just briefly describe the frugal innovations to develop more point of care based assays, or at least ones that can be done in rural labs. And also discuss a little bit about the intellectual property and how that will be shared. And there will be freedom to operate with the therapeutics that we're discussing today. Okay, so first I'll frame the question in Canada just very quickly. There's about 65,000 people living with HIV, which is, we're just slightly smaller in population size than Uganda, and you have about 1.5 million people with HIV. However, just like everywhere, we need lifelong treatment to maintain viral suppression. And unlike here, it's costing us about $30,000 a year to maintain individuals on treatment. Then when you add what Canada provides to the Global Fund, which is about $1 billion every 3 years, it's costing the Canadian taxpayers in their healthcare system about $1.5 billion a year. In Canada, it's a very interesting pandemic because it's not equally distributed. So, of course, we have our men who have sex with men populations that have the highest incident rates of HIV. But more recently, we've had a doubling and even tripling of new infections in our Indigenous communities. And so our rates in those communities are at par for just about everywhere in the world. So those incident rates are alarming. And it shows that even in our country, we sometimes can't control what's happening in transmission. And a lot of that is due to injectable drug use in those communities. So what we're trying to do is with Immune Equity, which is a program now to, as I described already, develop therapeutics that can be produced and utilized at the same time in both low and middle income countries and in high income countries, and the same therapies. What we're needing to do is integrate with our clinical trials unit. We need to establish GMP manufacturing in Canada, which is a real problem in Canada, not just in Uganda. In Canada, we have very few resources for that. And we need to do more bi-directional innovation with Uganda and to get the scalability we need. The impact is, of course, we can eliminate in Canada lifelong art. And that is projected to be anywhere between $30 and $45 billion in savings in the healthcare budget if we can enact a cure for HIV in Canada. And that's over a 20-year period. The savings in Uganda are at least to that extent or not more. And then it's just the clear, obvious thing about improving quality of life and reducing stigma as it is here. And for us, it is also very important, as it is here, because that ability to cure HIV is a significant milestone around the world. And if we're able to do that and do it homegrown with our technologies, that'll advance Canadian biotech and will put us in a global leadership position along with Uganda. So we're trying to develop a partnership with the JCRC to advance these affordable cures. Oops. Again, it's establishing a strong partnership. And the other thing is we have to come up with an approach that's systemic. That's systemic. And what I mean by that is we need to incorporate all aspects in care and delivery of the treatments, in community advisory boards and getting the community educated and involved from the get-go. And another big issue, as I mentioned before, is making sure that we have a pathway to share intellectual properties. The things that I'm going to describe of these therapeutics, they are licensed to other companies, but we've also had agreements with those companies that they're willing to share the IP for freedom to operate. We already know the challenges here, but there's one that I want to address because it's come to light more recently. And one of the things for an HIV cure that is essential is that individuals that are eligible for an HIV cure therapeutic are on stable antiretroviral treatment and their viral loads are completely undetectable. Without that, an HIV cure isn't really achievable. And it doesn't matter what type of cure you're implementing. And why I'm describing that now is because even though we have really good response rates in Uganda for the latest antiretroviral treatment, BTG-based therapies, and we may also even have a greater response rate when we go to long-lasting injectables. The problem is funding is unstable for those treatment programs. And what we've seen more recently is an alarming emergence of resistance when people do fail those treatments, which we hadn't anticipated. So I'll just overview that just quickly. It's not moving. Okay, so this is more recent data looking at treatment outcomes in Uganda in the national program that we did with the JCRC in a big collaboration. I should say it's led by the JCRC. And basically what we're seeing is with the introduction of DTG-based ART, the odds of viral suppression have increased at least fourfold. So that's remarkable. And the failure rate, the least virological failure rate on DTG-based ART is somewhere around 5%. It peaked at about 7% in about 2021, 22, but has actually been coming down since then. The alarming part though, aside from now the failure rate actually being reduced is that when individuals fail, the emergence of resistance to DTG is quite prominent. As compared to low to middling or high income countries, DTG resistance, especially in first line treatment, is very rare, even when you see failures. And what we're seeing just in the integrase gene alone, that we're approaching 25% having resistance. And this, when we consider other off-site mutations that appear outside of integrase that still confer resistance to DTG, it's somewhere near 40%. So this is higher than it's ever been reported. It's probably happening all over Africa, it's just not being studied. And that is problematic if there's a possibility of transmission of that DTG resistance. I just do it here. Okay, I'll try to keep going faster, so here it's gonna stop. So we have a lot of challenges, economic burden, we won't go into this, but it's quite clear that we need a cure. Okay, so the ideas here are to incorporate affordable cure development in its development of these various therapeutics, and I'll outline 3 specific therapeutics that we're pursuing. And then the frugal biomedical innovations, which I'll outline as well. And then this other aspect of it, of being especially cognizant of equity and inclusion. In Uganda, the diversity is largely people from Bantu tribes that populate Uganda. But in Canada, it is a huge diverse population. And one of the difficulties is our immigrant community, especially the Black Africans and Caribbeans, are not getting the same equity in care. So it's along with the indigenous populations. And we just need to build that trust in our communities, and you will already establish that trust largely in your communities. So the way we approach these cures is a basic kick and kill strategy. What that means is we need to find a way when a patient is on treatment that have very low to undetectable viremia, to tease out the remaining viruses out of their reservoir and then somehow eliminate that virus that's been teased out and to kill. And we're going to do that with our initial products, which will be this HIV-like particle, which has been shown now probably to be most effective in driving out latent HIV in a very targeted manner, as opposed to hitting all cells, like most of the chemotherapeutic agents that have been used thus far. And then using these stem cell derived plasma cells to produce broadly neutralizing antibodies, and to also enhance this with a CAR-NK cell, which I won't get into any detail in this presentation because CAR-NK cells require a whole 45 minutes to describe, so it's too hard. But the important thing to note is all of these are allogeneic, so they are not autologous. They don't need to be personalized approaches. They can be produced anywhere in the world, and they can be provided almost to anyone. And there's really not much of an opportunity for stem versus host, graft versus host disease in these type of transfusions, in particular with the plasma cells, because they don't have the class 1, class 2 proteins that often relate to that graft versus host disease. So this is really important because most of the approaches that are being utilized today are mostly autologous. And to give you an example, we cured a patient in Toronto recently, but that requires a matched donor to a certain extent that has a CCR5 deletion that makes that individual resistant. That individual had a leukemia, non-Hodgkin's lymphoma, and were being treated for non-Hodgkin's lymphoma with a transplant and complete myeloablation. And as you know, that's very risky in and of itself, but then to use it to also cure an individual is an advantage, but it's not something you're going to do. But one of the things that is never discussed is the cost. So the cost of that treatment, even in Canada, is $350,000 per individual, and it's not affordable, even at a $30,000 a year cost to provide a patient treatment. It doesn't add up, and our healthcare budget would just be blown out of the water with that kind of types of treatment. Okay, so I'll talk a little bit about HLP and VNAPs and how they work, because it's kind of important to understand how we produce them and why we're approaching these. So I'm going to teach you immunology in about 5 minutes, at least in regards to this. So just pay attention and you can say you know immunology, okay? Okay, so to go back, we have someone transmitting the virus to another individual. What happens in that case, the virus gets into your system. We have these cells called dendritic cells that pulls the virus out of your system, and then it carries the virus to your primary lymphoid organs, like the lymph node. And there the virus gets presented to T cells primarily as a first contact. And these are CD4 positive T cells or CD8 positive T cells, but it will just stick to CD4 positive T cells. Okay, and that antigen presentation leads to an immune response. And that's what allows us to mount a response to viruses. Doesn't matter what antigen it is. Okay, and when that happens, in that presentation of that antigen to the CD4 positive T cell, what it does is it tells the CD4 positive T cell, you got to start thinking about killing this antigen, okay, the whole immune system. So what it does is it activates it. The T cell is now ready to deal with it. And it's going to do a lot of things in the immune system, but it activates them itself first. And it also proliferates, makes more copies of itself, because these cells are specific to HIV as an antigen. So these ones aren't going to recognize other things, just HIV. And they're going to go after the virus. And this is a continual process. So it just keeps going on and on and on, as long as HIV is present as an antigen. When HIV gets eliminated, this stuff shuts down, the T cells go into cell death. And if they didn't, all of the HIV patients would be basically walking around with leukemias. And if it doesn't happen in any individual, you'd all be walking around with leukemias. So it's really required to eliminate the pathogen to basically resolve, and then the immune system is ready for the next challenge. And so what you have in the lymph node is you have viruses being presented as antigen in this germinal center. And it's just in this, between this light and dark zone. Okay, that's not important. What the problem is with HIV, and this has been known since HIV has been discovered, and we found out its receptor to enter cells is CD4. So that means that not only does HIV induce an immune response like every antigen does, but HIV also infects the same cell that's trying to protect us. And in doing so, HIV is replicating at high levels in the CD4 positive T cells that are recognizing HIV as an antigen. And HIV can only replicate in CD4 positive T cells that are activated. So all those CD4 positive T cells that are sitting around ready to encounter another antigen, they're not activated, they're just sitting quiescent, quiet. And so this is one of the things that is paramount to our approach. So I'll just go ahead. So what happens then is you're creating more virus, more antigen, more CD4 positive T cells are getting activated. And that virus is infecting those same cells and it's just churning. And until you add treatment, you're not shutting down that process. And most of those cells that are producing virus die. But some of those cells become memory cells, and those memory cells are important that if you re-encounter an antigen or another a virus, normally it's giving you a recall response so that you can combat the flu after you've been vaccinated, for example. But in this case, what happens is that memory T cell often gets caught with a latent HIV. Same thing with Epstein-Barr virus, CMV, a lot of different, especially herpes viruses, do the same thing. And that can be reactivated later, and when it's reactivated, you get HIV all over again. Okay? Now, normally, this virus is constantly churning, so it's constantly reactivating the latently infected cells. So the long-lived memory T cells only appear when you start treatment. Okay? So they then stick around. They stick around for years and years and years, and they never go away. So as a consequence, when you take away treatment, if you stop antiretroviral treatment at any time, what you see in those red lines is, is that the virus rebounds within a matter of a week or two. So basically you have to keep the patient on lifelong treatment. Okay, so everybody got that, right? We're finished immunology 101. So what we do is we take HIV that is what we call an HIV-like particle. It's identical to HIV, although we've injured it so much that it cannot replicate. It is completely dead. But in many cases, it represents the population of viruses that exist, let's say in Uganda. We have a version that's specific for Uganda. We have a version that's specific for the high countries. And when we provide it as a potential kind of like immunization, what it does is it awakens the recall responses. So now all those T cells that recognize HIV in an antigen and were sitting back in memory suddenly get turned on. So there's two outcomes to that. One is you induce an HIV specific immune response, which is great and it's immediate. The second thing is you're also activating those cells that have latent HIV in them. And those cells are very hard to activate because they're at very, very low abundance. And so in order to activate them, you have to find them. And the best way to find them is with HIV. So a dead virus particle teases them out, regular antigen response, and it gets rid of most of the latent HIV that's present. And then with continual treatment, you kill off all the viruses that is being produced. But then the HIV specific immune response also helps to deal with this infected cells to kill them. This response can be boosted, and the way we boost it is by using broadly neutralizing antibodies primarily. So that'll help remove anything that the antiretroviral treatment can't remove in the short term. Okay, last slide on that on HLP, and we'll jump to the next one. But HLP, all of these drugs shown here, verinostat, romidepsin, babinostat, priostatin, these are all drugs that have been used in clinical trials. They were originally developed as chemotherapeutic agents for cancers. And they're just off the shelf drugs in clinical trials for HIV cure. Their ability to induce latency reversal is relatively good, but they hit every possible cell that is in any type of state of ability to be activated. And what that means is when you're trying to go for that needle in the haystack, where it's a 0.1% of the CD4 positive T cells that have the latent HIV, that sort of pleiotropic effect is really damaging and they're highly toxic. So HLP, the difference here is about 1,000 times more active. And it only is activating those cells that recognize HIV as an antigen. So it's inducing almost all of the latent HIV, and that latent HIV appears to be in those HIV specific T cells. Oh, the other point I'll make. So I won't go back, it's okay. This is a very easy product to produce. It's a biotherapeutic, we can produce it at mass scale. And GMP processes. So it's unlike a chemical drug, it's very easy to make.
Let me ask you.
Yeah.
But this, if we just go back to that, I just want to make a point to maybe here. The potency of HLP as a latent reversal agent is obviously very high.
Number 20. Thanks.
Yes. And the way you describe it, it's just like HIV, but it's being deactivated, but still has the potential to stimulate immune cells in the body. So would we need to evaluate this product in animal studies before we come to a patient? Because that shortens or elongates the process of going to the clinic.
Yeah, so right now we have 2 cohort studies in mice and humanized mice studies that we're testing it. And one is halfway through, we don't have the results, the other one just started. And we're using it in combination with the new VIV latency reversal agent, which is a SMAC inhibitor, inhibitor of apoptosis inhibitor. So, but VIV doesn't exist anymore, so it's GSK. That we're moving forward with on the animal studies. The problem is, and that's what we've discussed with numerous people. This particular product can go first in human, probably more effectively than many, and avoid macaque use, because the macaque studies will take so long and are so expensive to do. The other thing is it's highly dependent on, in this case, the HLA alleles that are presenting the antigens. So in macaques, of course, they don't have HLA alleles, but they have their macaque leukocyte antigen alleles for +2. And that changes things because they're all kind of inbred. And so for the most part, what we have is a very similar antigen presentation during the trial. I've tried to get Gates Foundation. They were originally going to fund macaque studies, but because our toxicity studies show that there's no toxicity, that I've been told just to go for first in human. And so that's the way we're proceeding right now. The other thing is that there have been studies like this in HIV, people living with HIV and on treatment previously. So it's not uncommon to give them dead virus. for various types of studies, or even give them live virus, which has been done in a couple Spanish trials. So the outcomes for safety were fine. I wouldn't propose to give anyone a live virus, but
But my question is also targeted to how we can move fast
yeah,
with a product that is available. And I appreciate the background, but maybe these are things now we have to start presenting to our regulatory authorities in terms of this is why we want to move like this. And so I know I was part of the proposal writing and we have this 6 years. I don't know that we have to wait.
no.
Yes.
Yeah, so the way, I don't think it's advisable from a optics perspective that we need to stagger phase 1 clinical trials. If we do them in Uganda first, like right off the bat, then it'll be problematic. I mean, because they're saying, well, you don't want to try it in your own population first. And I don't think we want to go in that direction, but I don't think that's a problem. I mean, I think we're on track to go to phase 1, I'd say in a year and a half, because the only problem we have right now is finding a place for production. And that's why we want HARP available. We have a GMP production facility being built for biotherapeutics, and it just makes more sense than to contract out to an outside contract research organization. So that's really what's holding us back right now. Yeah. I should mention, this is a little bit, these are all samples from individuals that have been on treatment for years, and this is what we can see driving out their latent HIV. The other thing is half of these participants were from Uganda. So we saw no differences in Ugandan versus Canadian sort of outcomes in this analysis. Okay, so let's move to this. All right, so I'm not going to go through antibody responses. I just want to put this up to tell you that right here, those plasma cells at the bottom of this slide, these are derived from B cells and they produce antibodies. So when you drive your immune system to produce antibodies, that's the pathway it follows. And those are the antibodies that are produced from plasma cells. Plasma cells are stable. Again, they lack a lot of the proteins that result in non-self recognition. And so they can be easily transfused into individuals without graft versus host disease that survive for a very, very long time producing antibodies. The difficulty here is they would be producing antibodies that we don't want. So my colleague, Peter Zanstra at UBC, who's a leader in cell-based therapies and production of cells for cell-based therapies. He's developed a system to go from hematopoietic pluripotent stem cells to derived plasma cells. And so that is well established. That's the protocol. I'm not going to go into detail, of course. But the other thing that's important is, again, those plasma cells are not making the antibodies we want. He has done a lot of gene editing, and so recently he's successfully put in a broadly neutralizing antibody into plasma cells. That broadly neutralizing antibody inhibits most, if not all, strains of HIV. So, and he's shown now that it is producing those antibodies and that can inhibit HIV. So that's the kill step. That's an enhanced kill aside from just antiretroviral drugs. And those can be mass produced, and he produces them at mass already for various other applications. I should mention the HLP is with a company called SHC Therapeutics, and this is with a company called A3. Biotherapeutics, and both of them have agreed to allow for the IP to be transferred to Uganda and for Africa.
Don't understand the implication of that. For IP to be transferred. Yes.
Yeah. Yeah, I mean, I think every company should do that, and Gates
But they
Foundation
don't.
has, I know, but it is remarkable. Because honestly, the market in low income countries to make money is minimal. And so all you're doing is preventing people who need it from getting the drugs. When it's also necessary in high income countries where people will pay for it and healthcare systems will pay for it. So it's a win-win. It makes no sense that it wouldn't be done. So. Okay, so I mentioned already where to produce it. This is a new building we're building on campus called Pathogen Research Center. About half of the space in the Pathogen Research Center is a GMP clean facility. But it's specifically designed only for things like vaccine, antibodies, DLPs, all of these other products that are a therapeutic, but it's a biological. So, and if you look at most modern therapies that are being used in most diseases these days, it's, I'd say 80% antibody based. So this is what this facility can be used for any disease. It doesn't matter. It doesn't have to be an infectious disease. But one of the things is would be to produce HLP through this facility for these processes. So a major part of this grant is to replicate at least part of those production capabilities here in Uganda. And that is for, in large part, cell-based therapies, but also HLP, because HLP is basically a cell-based therapy. You use cells to make the virus and then you treat the virus. In both situations, your endpoint is not the cells you're providing, but it's the antibodies or the HLPs that are doing the business. And I won't again talk about CAR T cells, but our CAR NK cells, but they could do the same thing. So we want to have this constant exchange of information flow in the development of the production processes, and then try to frugalize or reduce the cost here as much as possible to manage production so it can be utilized for therapy. Okay, so the last thing I'll mention is the idea of frugal innovations for treatment monitoring. So you realize that when you look at an individual on HIV therapy, we only do viral load monitoring once a year in Uganda, right? And that is generally sufficient. I might argue that the recent problems we've had with DTG-based treatment and the emergence of resistance is because we're only monitoring once a year. So that can delay treatment regimen changes sometimes a year and a half, 2 years later, and that has resulted in problems. But in an HIV cure implementation, you need to monitor HIV treatment at least when you start the initiation of that treatment, at least every 2 or 3 days. And it doesn't need to be an accurate determination of viral load like we've been doing, but it has to be, I think, a plus minus situation. So you need a way to quantify how much virus is coming out. And it's okay if there's little amounts of virus that emerge, because that's what you want to do, is drive it out. But as soon as you start seeing a viral rebound into the thousands, tens of thousands, and 100,000s per ml of blood, then you've got issues and you have to put them back on treatment. And you can't wait a year for that. So you need to monitor very frequently, and then eventually you can stop it and go to a slower schedule of treatment monitoring. What that means is that the national labs cannot possibly handle that. I mean, it has to be relatively rural clinics point of care in order for this to work. And right now, Uganda, we are not set up for that, right? We can do it, but we need cheap assays. So that is what the Frugal Innovation Platform is, and that's to do real-time PCR-based assays, but with the cheapest thermocyclers you can find. Okay, so we can just put them in clinics, put them all in the JCRC centers, even get them into more rural clinics, and just simple, no extraction PCRs from whole blood, and then have a fluorescent monitoring system. And this is what David Holsworth, a professor at West. So a real-time PCR machine's costing about $60,000. For an effective one. And that's, a lab can't afford it. I mean, we have them at GCRC, we can afford it, but rural clinics can't afford it. Whereas you can afford a thermocycler. Thermocyclers are now relatively cheap. You can almost buy them for about $1,000 or less even. And what David has developed is an optical device that basically just has a little chip sensor that will read the fluorescent signal and do what a real-time PCR machine does, but you can just put it on top of a thermocycler. And this is just his prototype. They've now done it for 48 well thermocycler, and now just like normal, computer-based systems, you're just shrinking it so you can put it on a 96 or even after that 384 systems, so that you can utilize it either at scale or even in a 24 well system when you're at a rural clinic and you might not have to do it as frequently. And this is kind of the approach. You just put it on top of the system and you monitor it in this circumstance. And he's managed to even detect the fluorescent signals. So it's working quite well. So they're trying these other frugal approaches that would be specific for implementation on HIV cure therapeutics. Okay, we're not going to have an ability to solve all of the diagnostic high cost systems that we face in biomedicine, but we can focus on the ones we need for this project. And that's the team that's working on it. They have partnerships with both Kenya and Uganda, and so there's a team of Frugal Biomedical Innovations in mostly at Must and at Macquarie. So they're working. Okay, last couple of slides is how will we do this? This is a very confusing slide, but I just wanted to show you the sort of the intricacies of what we're discussing. So there's a few components that are critical. One is training. So one of the things that we want to do even before this grant starts is I plan to send someone over from my lab that can provide the training for HLP utilization so that you can do it here on any sample tube blank from patients, so we can see the effectiveness here. So that will be done possibly this year. The other thing we need to do is we do have the HLPs made for the different HIV subtypes that are present in sub-Saharan Africa, but we haven't really tested them to the same extent that we did with the subtype B, which is for North America and Europe. So that's something that we would do as along with the training. It's not really that difficult because protocols are well established. At the same time we're doing that, we're basically looking at moving the current HLPs into toxicity testing, and this is all within the first year. They're all ready to go for toxicity testing, and shortly after we'll use the HLPs of the other subtypes to go into toxicity testing as well. So that puts us within year 3 that we could start under this guideline, a phase 1 clinical trials. And I would say staggered about 6 months later, a phase 1 clinical trial in Uganda. These are really optimistic predictions, but I mean, if you're proposing it as a grant, you gotta look optimistic. And then you follow that through. Post-year grants will probably be in the later phase 2B3 trials, but that's kind of where we're at. The plasma, the stem cells derive the plasma cells with the broadly neutralizing antibodies. Those are still in preclinical development, unlike HLP. And we're looking at finishing that preclinical development, also transferring the training, but we need the bioreactors for production here. And they already have them established in Toronto and UBC. So that's going to be the major thing. And Western has been willing to invest some of their indirects to provide at least that initial scale production that you need in Uganda. So that's one of the things that we look forward to. And then it just follows on from there. The last thing I want to mention is there's a whole layer of law involved in this and all the IP transfers to make sure this can go smoothly. And there's a team of people that are involved in patent laws that are going to be working on this. There's an economic assessment to make sure that whatever we're proposing to do is cost effective, because as soon as we move out of that cost effective realm, it doesn't make any difference how good it is. So we want to make sure this is cheap. So we have a team doing the economic models. And I already mentioned just the production and the frugal biomedical. Last thing is that most superchargers we're going to have is leading our community advisory board here. And then we have a partner for him in Canada. And we have this cool idea of having our community advisory groups meet frequently together. And so there's this thought that Canada's getting something better than Uganda, and this appreciation that we're in it together that I hope we can foster. The other thing too is we are not stupid in the sense that this funding that we might get is going to be sufficient for clinical trials, but we think we can kick it off. And we're looking at a lot of different VCs through the companies, the biotech companies that are involved to try to get funding. The other thing is, I think both Sissy and I really feel that if this really works and we get sufficient funding, we can convince Mike McCune and the Gates Foundation to finally put his money where his mouth is and give us funding that can help. But aside from that, there are other potential donors that can continue to fund this, at least I hope. And I think that's it for me. The role of the JCRC is quite obvious. I can't think of a better institution that's done more clinical trials in Africa on HIV that can pull this off, and that has an interest in cell-based therapies and gene therapies, which is what we need. Again, I've mentioned already that we're very much about making sure that this is focused on providing treatments for people that need it the most in low income settings, and where resistance and failure on current treatments might be problematic. And I won't go through this. There is a large economic potential for this. We can't ignore that. I mean, the market is somewhere around, if let's say you get full penetration into the market for an HIV cure, displacing HIV antiretroviral treatment, it's somewhere at a minimum of about 10 billion a year. So the people who are shooting for a cure realize the economic potential. And there's no question that even its development in sub-Saharan Africa will provide economic growth for this region as well. So that's something else to consider, job growth, biotech development, etc. And we'll end it there.
Thank you very much, Eric. We've just finished a retreat looking at our next 5-year strategic plan. And immune equity is the central focus for our HIV cure program.
Yeah.
So we had better get that money.
No pressure, eh?
No question. So I have 2 questions. They already comment. One of them is what we have seen happening in the sickle cell disease space. There were several companies working on ex vivo gene therapy. And maybe up to about 5 pulled back and stopped developing their products because they were products that were already available. For HIV cure, now, of course, there are many other people working on it and many strategies. So, and that's why I was asking the first question. There is already work that has been done and we need to move fast. So that we are ready, especially for non-developing countries. Yesterday, again, I was writing the strategic brief for the sickle cell disease, and Gates Foundation is funding Tessera, which has a gene writing product for sickle cell disease. It's doing very well. But it's also now getting money from APA is basically AR Generon, over $250 million to also develop an HIV cure using the same gene writing concept. But I think we are just starting. So my comment there is we should go as fast as possible. Even though we do not have $250 million.
Yeah, you don't have that. No, I agree. I mean, we have to maintain a certain generic aspect to this. If something else comes along, that especially biotherapeutic like gene editing, you can adapt to it if need be. And I'm the person to say that I cannot guarantee that a combination of HLP and a stable production of broadly neutralizing antibodies is going to provide a cure. But so far it's still looking the best. I mean, the problems with gene editing is you can gene edit for an allogeneic transplant so that you can get cells in that will be infected. But you can't do that to cells that are already present in your body, right? It's like there's no targeting mechanism unless they use something similar to what we're doing.
Yeah, and I was just thinking about it because the reservoirs and where they are inserted, there are different sizes and different length, so which live RNA are you going to use to get all the reservoirs? So yeah.
Trying to think of the company, but there was a company that was approaching it like that to kind of like target the integrants of HIV, and they went bankrupt because they thought they had a product and most of their data was shoddy at best. So, I mean, the problem is just that, like, how do you get at less than 0.1% of the T cells in lymph nodes?
other questions to which you may not have an answer, but what is our target in terms of cost of this combination for low and middle income countries?
Back of the envelope, like, I think it's gonna be at least a couple $1,000s per individual. The therapies might run 1,000, let's say, and then the monitoring, if you can get the cost down, would probably be at least 1,000 as well. So it might be as much as 5,000, I don't know. It all depends on production, and right now it's the process of producing is not difficult, but it's the amount you can make is what I'm concerned about. So, I mean, we have other approaches that we might use to produce it. One of it is to produce it sort of constantly in an individual through like a viral vector like VSV, and you can produce the viral proteins that make a virus particle and you get them released from cells. There's other ideas, but what I prefer is just to make them as VLPs, HLP, and just produce it as a therapeutic, that you don't have to have a vector to produce it. And part of it is my ignorance in not being involved in that production, and neither will I be, because it's not my area. So I have to trust the people who would make it at large scale.
Okay, so I just have to jump into the STP poll. It wasn't showing on my calendar, but it's starting now. And I will leave the team, since I'm more conversant with what has been happening, and we are waiting for June 2nd to know if we are invited. There is a phase of interview, and if we are invited for interview, I think we will be in.
What's that? We've been.
For the interview, for the new.
Yeah.
Offer.
Yeah, so that's one thing that's problematic, me leaving early, and I might need the help of the JCRC. We need video footage of stuff going on in the clinic, the labs, just little snippets that we can parse together. Maybe like 3 or 4 minute interviews with senior staff on video. Again, we don't, I think our video for the presentation will run maybe 5 or 6 minutes, because we only have 10 minutes to present before the test should start. But we thought a video presentation would be a hell of a lot better than, for example, the slides that I presented today, which are not that great, by the way. So I might, Nelson, I might need you to help or someone, yeah. Oh, sure. What helps the T cells only identify and kill in HIV infected cells? So once the latent HIV, let's say, is released, if this is related to the question, it's going to be expressed on the surface of the cells, the proteins of HIV, and they can be targeted by cell-mediated response, cytotoxic T lymphocyte responses for the killing. And that would destroy the latently infected cell that's been reactivated. That's one thing. There's also this ADCC, which is a similar cytotoxic T cell response, but mediated through antibodies. And the broadly neutralizing antibodies would do that. The tricky thing, and now we're getting into the weeds, is that a viral protein called NEF, it downregulates antigen presentation on the surface of the cells. So it avoids immune recognition. So we have these, it's important to know that we have. Like the next phase of development of products, so we have HLPs. That will deliver things that will block NEF from doing this. And we have HIVs that will also, HLPs that will further stimulate through our through RNA adjuvants, TLR ligands. So we're testing those now, they've all been developed. So that's the thing is, like, with any product development, you don't want to stop at one. In what you're doing, so that you make sure that, you know, you've got something better to try it the first time it doesn't work as well as you hope. What is the half-life of the HLPs? How many planned shots will be given to ensure clearance of the latent virus? That's a good question. We're trying to do that in mice right now to see, especially it uses the same pathways as antigen presentation, so like with a vaccine. The difference here is with a vaccine, you just need to stimulate the response. With HLP, we have to hit basically most primary and secondary lymphoid tissue. So that's why it will require, we estimate about 4 immunizations and then spread out sufficiently so that the immune response doesn't basically kill off the HLP. So you want to dampen the HIV specific immune response before you provide the next round of treatment. So it's probably 4 injections spread over a period of about a year or less. And each time we hope that it's a diminishing return of the virus, so that by the time you get the last shot, there's almost no latent HIV there, but time will tell. Okay, the other question is, study estimates half-life of latent HIV to be about 44 months in people on continuous ART. 3 to 5, yes, absolutely. It depends on whose study you're talking about, but yeah, half-life, sure. But what we've saw with our studies, and other people have seen this too, is you can get someone 25 years on treatment, you remove treatment, even ex vivo, and their viruses will rebound. So there's estimates that to eliminate the latent HIV reservoir, in many patients, it could be as much as 70 years before HIV is no longer there. And honestly, I think it's one of those things where you have these spontaneous, almost clonal expansion of memory T cells. It's almost necessary so that you keep a memory T cell response. And that's why we can get vaccinated with, for some viruses or diseases, once in a lifetime, and some we need to do it more frequently. And that's largely related to how long the memory cells live for. So right now, like most people will not live as long as their latent HIV reservoir could live for. So most people will die of other consequences or old age before their virus is cleared. All right, that's the 3 questions.
There's one more here.
Oh, okay. That was are there underpinned response biomarkers for which biosimilar companion diagnostics could be developed? Yeah, so there's kind of 2 things to that. So the one thing is, there's been a large search on and lots of good published papers on trying to find biomarkers that would identify the cells that contain latent HIV. And you can kind of do it, but it's kind of like not one biomarker. It's a combination of a lot of biomarkers that can kind of tell you that there's latent HIV there, but their false negative rate is very high. So it's problematic. And for the same from the same respect, it's really hard to use those biomarkers to look at. If you still have latent HIV left in your system, or to use it to monitor the effectiveness of a cure. So far, there's no solid biomarker, with the exception that a T cell receptor that recognizes an HIV antigen presented by class 2 is the sort of biomarker that really defines whether a cell has latent HIV or has a good proportion of those cells have latent HIV. And then T cell receptors that recognize other viral antigens or other antigens tend not have latent HIV in them. So you could use that. The problem is the diversity of T cell receptors that recognize HIV antigens is huge, and we haven't defined that very well yet. Hi, that's Fred. Oh no, this is Fred. Okay. Hi, Eric. Would there be any mutations that would prevent it from working well, such as different cells infected with HIV? So, yeah, Fred, what I didn't describe in this talk is that we're developing a bioinformatic analysis tool. And that is, we suspect that some people that have specific HLA class 2 antigens may not respond to the HLP effectively or not sufficiently for a cure. We estimate that might be as much as like 10%. So we're developing this algorithm where we can sequence the HLA class 2 alleles, and we can use this predictive algorithm of what HIV antigens they would present and whether those HIV antigens are present in our HLP. And in those conditions where there's not an effective match, then we're going to have to make an autologous, or we'll have other stocks of HLP pre-made to deal with those individuals. So we don't know exactly yet, we're working on it, but it might be as high as 10%. That's what Joshua Jalova is working on in the lab right now. And is it as in different infected cells? So one of the big questions here is it only CD4 positive T cells that have latent HIV? Probably not. There's probably some myeloid cells that have latent HIV, but the fractions of those are very, very small compared to the CD4 positive T cells that have latent HIV. And we suspect that you will need other latency reversal agents, possibly, to flush those out. So that's why we're testing this with other latency reversal agents. And the other thing is my own opinion. I don't think that latent HIV in the myeloid cell population is going to be difficult to control if you've induced an HIV-specific immune response. And with treatment ongoing and HIV-specific immune response before you remove the treatment, that might be sufficient to flush that latent HIV out. All right. Fred, I miss you, man. I can't believe you're over there and not here.
Yes, Eric. Thank you for the presentation.
Okay. Next time I come back, I'll be here. And hopefully Ebola won't be here, Fred.
Let me wait for Ebola to go down. I come back.
Yeah, well, you better watch it because they might let you back in the US. All right, say hello to Immaculate. I'm working on the paper.
Okay, I will.
I would give you the grant.
What's that?
I would give you the grant.
Yeah.
Oh, nice. If it was only you deciding.
So thank you very much, Eric, for the wonderful presentation.
Thanks, Nelson. Okay, we gotta get students back here, because we got work to do, we need people to do it, and. I'll send around a couple of questions, like, yeah, it's not necessarily questions, we just need. Like, let's say if it's.